Hello. Good morning all. Are you all back after the break? I think you had a long break, is it? Or a short break? Okay. I hope all of you are fine and doing great for today. And I'm happy to be with you again. And I hope it's the same the other end. Okay. I'm always happy to be with you guys. Before I start with, I have mentioned about completing the notes, and I am getting people on my personal window. They are sending me the notes. Don't worry, on my personal window there are a few of them giving up their names, and they are sending me the notes. Take your time, do it neatly and correctly. I mean, when you take it, because it's not directly from the board, it's, you have to use it's sent on your mobile or from the laptop. When you're copying it down, be very careful when you copy it down. Then I hope you have read till whatever I have taught till now. If you have doubts, please let me know. Sometimes at point, when you ask me down, maybe I may not be able to tell you then that, but immediately I will respond back to you. Then, so we have stopped yesterday. We have learned more about nucleus. Then I've told about the cytoplasm. You have to, I think by now, if those who have read would find it easier to understand whatever I've done yesterday. And I've told you, you can watch our recorded YouTube it will reinforce you. You will find it more easier once you are there in the live session. Sometimes some of you might get off because of the network connection problem. Don't worry, it is there in the YouTube. If at all any videos are not there in the YouTube, it will be uploaded soon. So don't get worried. I got yesterday some of you inquiring that video is not there, this video is. Don't panic. It will be uploaded. Maybe a little here and the technical issues, but it will be uploaded as soon as possible. We are trying our best to do that. So today we'll go. I said there are some cell organelles which are very important, and because they carry out very important crucial functions in cells, like the ones which I said, Endoplasmic Reticulum, Golgi Paratus, Lysosomes, Mitochondria and Plastids. These names are very much familiar by now to you all. So we will go today to our dear Endoplasmic Reticulum. Okay, Endoplasmic Reticulum, or what I always give a short form, ER. When you are going to write notes about this in your notebook, you have to learn to draw the picture also. That will be sent by me. I will send, because the pictures in the book is not that very clear and very few only have been given. So I will send those pictures. The picture, because it can be a question, explain endoplasmic reticulum with a neat labeled diagram. So you have to know that. So I will give that, nothing to worry. Whatever notes I've been updating, you have to update in your notebook. So here we will go to the next organelle, that is endoplasmic reticulum, or we can say ER. How it is, the structure of it. It's a large network of membrane-bound tubes and sheets. They look like long tubules or round oblong bags. Long bags, the height will be less. They look like long oblong bags, a form of bag, but it will be long. The height, it's less. Now it is similar, ER membrane, it has a membrane that is similar to the plasma membrane, similar structure to the plasma membrane. So how the structure is, it's a large network of membrane-bound tubes and sheets. You have to write it correctly whenever you are learn it correctly. They look like long tubules or round or oblong bags. Either of the word is enough. Either you can say long tubules or round oblong bags. They are called vesicles. Okay? They look like long tubules or round or oblong bags, and there's a word they're called vesicles. Now the ER membrane is similar to the plasma membrane. The membrane, it has an endoplasmic reticulum membrane, is similar in structure to the plasma membrane. Now, last year when I was teaching, I always used to say about all this. You have that, you have mitochondria, you have endoplasmic. There is 2 types of endoplasmic reticulum. There is like rough, smooth. I used to always mention so that I was making you prepare for this year. You will have such things. I hope you can see the image here. So there are 2 types of ER or endoplasmic reticulum. That is rough endoplasmic reticulum, RER. When you write in notes, please give the full form and you can give the bracket. Then smooth endoplasmic reticulum. So endoplasmic reticulum are of 2 types. That is rough endoplasmic reticulum and smooth endoplasmic reticulum. You can see the picture, how it is given. A rough endoplasmic reticulum. Again, the smooth endoplasmic. I'm sure you can see some differences there. There's a difference, and small slight differences there between both. And you can see the nucleus and the nuclear envelope. That is the nuclear membrane you can see. So the smooth endoplasmic reticulum and rough endoplasmic reticulum, network of long tubules. See, there's a network of long tubules. Now, RER or the rough endoplasmic reticulum, when you see them under a microscope, you find in the picture itself, you can see there are particles around it. Picture itself, you can see some particles around it. Now, what are they called? They're called ribosomes. This also I mentioned last year. Ribosomes. So, on the rough endoplasmic reticulum, when you watch them under a microscope, they have, when you see it under a microscope, it has particles on it called ribosomes attached to its surface. So rough endoplasmic reticulum, or they are rough because of these particles called ribosomes attached to the surface. Now ribosomes are present in all active cells and are the sites of protein manufacture. So ribosomes are present. Where are they present? They are present in all active cells. And what is the function? They are the sites of protein manufacture, or the protein synthesis takes place. So this is important. So rough RER is rough because they have particles on it called ribosomes attached to its surface. And ribosomes are present in all active cells, which are the sites of protein manufacture. Why I'm stressing in all this? Only then slowly it will enter your little heads. I can't rush on this part. You have to know each and every line in this. So you understood why RER is rough? Because of these particles called ribosomes. Now, the manufacture, whatever proteins have been manufactured or synthesis of protein took place, they are then sent to various places in the cell depending on the need of using the ER. So it is spread out, or it is sent to various places in the cell depending on the need using ER or the endoplasmic reticulum. This is important. Why it is rough? Because of ribosomes. And where are ribosomes present? They are present in all active cells and are the sites of protein manufacture. When I'm stressing this, there is importance for this. That is why I'm stressing and there itself. I know this chapter is not as you think it is. Every line is important in the chapter. Read the text. Underline the important points. Notes will be given. But at the same time, as I always say, read the text. Make a habit of reading text. Without reading the text, nothing will do good for you. Read the text. Just not the notebook. Teachers give a note. Read the text and underline important points. Now we have the SER, or the smooth endoplasmic reticulum. What does it help? It helps in the manufacture of fat molecules or lipids important for cell function. So here it helps in the manufacture of fat molecules. The other one is the protein thing. Here it helps in the manufacture of fat molecules or lipids, which is important for cell function. Now, some of these proteins and lipids help in the building of cell membrane. So the process in which the cell membrane or plasma membrane is formed with the help of proteins and lipids, a process, a name is given, membrane biogenesis. Underline, that's very important. Define membrane biogenesis will be asked. It's teacher's favorite. Okay. So that is membrane biogenesis. So ER helps, okay? It helps in the process called membrane biogenesis with the help of, they help with forming proteins and lipids. So membrane biogenesis is a process in which the formation of cell membrane takes place with the help of proteins and lipids. So whatever proteins are made here in this ER, some of these proteins and lipids help in the building of cell membrane. So this process of building up with the help of proteins and lipids is known as membrane biogenesis. I will give you the correct definition, but you must understand what it is. It's the formation of the process, membrane biogenesis, is the formation of the cell membrane, of the plasma membrane, with the help of these guys, the proteins and lipids, and ER plays an important role in this. Okay, the ER helps in this process, the membrane biogenesis. So RER, the ribosomes are important here, and the protein synthesis there. Here SER, the manufacture of fat molecules, lipids, which is important for cell function. Then I have mentioned about membrane biogenesis. This is also important. The correct definition I'll give you. You don't have to worry about it, but you should know what exactly it is. Now here they're just telling more about it. Some other proteins and lipids are there that function as enzymes and hormones. So ER also forms a network system. You can see from the picture itself, it is a network. So another set of proteins and lipids they function as enzymes and hormones. Lot of things are there now, it's going on in your head, I know. But please be attentive and be along with me. Now what is the main function of ER or the endoplasmic reticulum? They can ask you what is the main function of endoplasmic reticulum. What do? The main function of endoplasmic reticulum is to serve as channels for the transport, means to help or serve as channels for the transport of materials like proteins between various regions of cytoplasm or between the cytoplasm nucleus. You know where the cytoplasm is, or between the cytoplasm and the nucleus. So the main function, you can underline this if your text is open, or you can just note down this in your book. Or each, you must be wondering, there are lines in the, yes, I have been stressing on the lines from the text because the text is important. So the main function of endoplasmic reticulum is to serve as channels. Okay, the main function to serve as channels which helps for the transport of materials called proteins between various regions of the cytoplasm or between cytoplasm and the nucleus. So I hope you all are with me. Are you guys with me? Yes or no? Yes, I can hear somebody saying yes. Okay, thank you. One function we saw there, another function. Here, ER also functions as a cytoplasmic framework, providing a surface for many biochemical active cells. Here also, it functions as a cytoplasmic frame, the main cytoplasmic. Again, they are repeating the same thing, but a little more in a better way. They function as a cytoplasmic framework. Framework, you know what a framework is. A frame within the frame. They function as a cytoplasmic framework in which providing a surface for many biochemical activities of cell. Lot of activities happen in cell. So they play a great role in this as functioning as a cytoplasmic framework. Now, smooth SER, what do they do? The main role in the cell plays a crucial role, an important role is detoxifying many poisons and drugs, removal of poisons and drugs. Okay, they detoxify, they plays a great role in detoxifying many poisons and drugs, removal of the poisons and drugs in simple language when you say. So SER, this you have to see, you should not give this. RER, it is SER, smooth endoplasmic reticulum, plays a crucial role in detoxifying many poisons and drugs. Why I'm stressing each line? All the lines are important. Miss wondering, all the lines are there, all of them are important. So with that, we are done with the endoplasmic reticulum, the SER and the RER, the ribosome part. Okay, that is important. The protein synthesis, SER. I've given a main function of SER, I said, detoxifying or removal of poisons and drugs. You have to know all that. The function means what role they will see in this portion. they will ask you explain endoplasmic reticulum with a diagram. Or now we go to the next cell organ, the Golgi Apparatus or Golgi bodies, we can say Golgi Paratus. So they will ask you to explain it with a diagram. That is mainly in bio, we have the diagram stressed. So I will send that with the notes. So you don't have to, not most of the diagrams are there in the text. So each organelle, cell organelle, I will send the diagram. I'll try to make it easier because it's not like how we are seeing on the board when you're in the class. I'll try to send easy diagrams. So, but you have to know to draw them and to label them too. So don't worry. So now we will go to Golgi Paratus, the next cell organelle. This cell organelle got its name from a great scientist, Camillo Golgi. He himself gave this name. That is why we say Camillo Golgi, Golgi Paratus. He discovered it, so he, very smart of him, he gave his name. So it's easier to remember also. So Golgi Paratus was found or discovered or described by Camillo Golgi. So that is why they have been given the name Golgi Paratus. So this is another scientist. Remember we went in the beginning with a lot of scientists, discovered that, discovered the cell, discovered free living cells, discovered the RDR Virchow, the Sheldon Shimon, okay, then Lou and Hawk. I think all are giving me that smile now, okay. And yes, I can see some sweet smiles coming with all those scientists' names. I can see somebody grinning also, especially when that Virchow and names like that, Lou and Hawk, Sheldon, Shimon, and teacher is giving, and you don't worry about pronouncing. That is the way to pronounce, to tell you the truth. But some of you will be trying at home and then laughing and grinning. I can see faces and giving them, when you hear Virchow, who's Virchow? It is like that. We have to pronounce, so don't worry. Now we have another great scientist, Camillo Golgi. Okay, you can pronounce however you want as long as you have the right spelling. I can see somebody laughing to glory. I can see even the teeth, they're laughing to glory. That's okay. So now we'll go back, we come back to the portion. The Golgi Paratus described by Camillo Golgi. Now the structure, again, the structure. See, everything looks somewhat similar, but in a way they all are different from each other. But activities, all they are in this cell itself. They're doing all the activities, function, all the biochemical activities is done in cell. But all are different. They have different. I mentioned there are different cell organelles, and they play a great role in their functions. Now, Golgi Paratus, described by Camillo Golgi, what does it? Consists of a system of membrane-bound vesicles. They are flattened sacs. If membrane-bound vesicles does not come, you just remember, they consist of flattened sacs. How it is arranged? They are arranged approximately parallel to each other in stacks called cisterns. Okay? I want you to be attentive in my class. I see some of you playing unnecessarily, so be attentive here. I told you, we can see what you're doing. So be careful. Just joking, guys. So we'll come back here. The Golgi Paratus, described by Camillo Golgi. What is the structure? It's a system of membrane-bound vesicles we came across in the early portion. In brackets, flattened sacs. So they are flattened sacs. And how are they arranged? They are arranged parallel to each other in stacks called cisterns. So you must understand, when I've highlighted in red, it is important. See, these are the words will come to your mind. You will never forget. I assure you will never forget, because this is there in your mind always. Even in your sleep, if I ask you, what is the structure of Golgi Paratus, you will give. Children, please don't annotate. That means you're not attentive, you're just trying to play. Okay, we have done with here. You can see here the Golgi Apparatus, flattened sacs, okay, membrane-bound vesicles. You can see they are arranged parallelly. I hope the image is clear for you. I will send you the diagram of this. You don't have to worry. Some of you must be wondering, God, I have to draw like this? No, don't worry. It will be given a simple way. Now, these membranes often have connections. These membranes are RDR Golgi Paratus. They are connected with whom? The membranes of ER, and they form a part of complex cell membrane network. Okay? These membranes, the membranes from Golgi Paratus, they have connections with whom? with our dear ER, the membranes of ER, and they form part of the complex cellular membrane network. So they have connections with each other. So and they form the complex cellular membrane network. I hope the picture, this image which I posted is, you can see at least. Maybe the labelings, I don't know, cisterns. Okay, when two are called cisternae, that will be given in your book when I send you the diagram. So you can see how flattened sacs. Okay, let's move on. Here again, they're telling about the function as we have mentioned. The material synthesized near ER is packaged and dispatched to various targets inside and outside the cell through Golgi Paratus. So whatever material is synthesized or made here in near ER, I told you they have connections, right? So whatever is material synthesized, so whatever like a protein, anything synthesized there near ER is packaged and dispatched. Packaged and dispatched like in the postal service. Packed and it is dispatched. You pack and you dispatch, the same thing. So that will be easier for you to understand. I can relate that. In the post office, what is you? They pack it and they dispatch it. The same concept is happening here. And to where? To various targets, to various places of the cell, inside and outside the cell through our dear Golgi Paratus. Hope you understood. Why I gave you that example? You must be, oh my God, packed and dispatched. Now what is that? As I said, in the post office, you have those guys. They get the thing, chuck, they stamp it, pack it, and there it goes for the dispatch. So that is where the same thing is happening here. So I hope now you understood. Okay, see, if we were there in the class, it would be more rocking with the class. But as I told you, there are limitations, and I'm trying my best to be with you and to make the class more interesting. Then, the main function of Golgi Paratus. What is the main function of the Golgi Paratus? Its functions include storage, modifies, and packaging of products in vesicles. This is important. The main function of Golgi Apparatus. So here I said, whatever material is synthesized near whom? Near the ER is packaged and dispatched to various targets in and outside the cell through the Golgi Apparatus. I hope you got me. See, these are new things to you. So I will stress on sentences and words. Not that some of you will say, oh God, no, you have to bear with me. There's no other alternative. Okay? And then what is the function? I know you all are with me. Because I am getting feedback from you guys on my personal window that you are happy with me. I hope so. Okay. It functions, what are the main functions? Storage, modification, and packaging of products in vesicles. Here another function also is there. These functions are important. Okay, when I say that, the first one we learned, the storage, packaging, that one. Now we have here another function of Golgi Paratus is to make complex sugars from simple sugars. So they're made to make complex sugars from the simple sugars. So it has this function and the main important function, that is, it is involved in the formation of lysosomes. We have that next. Okay, Golgi Apparatus is involved in the formation of lysosomes. Here I said it functions, the first function, it functions include the storage, modification and packaging of products in vesicles, one. Then I said its functions to make complex sugar from simple sugars. Then I said the next function is Golgi Paratus is involved in the formation of lysosomes. Three functions are important. You cannot leave anything. You have to learn this. So I'm trying to make it easier to understand. That is that dispatch and package, same what that I gave an example so that you remember it. Now comes our dear lysosomes. Okay, you can see the picture. I'm sure it must be the picture in the book also. If not, don't worry. This will be given, drawn, made it easier with the labelings. Here, we said Golgi Apparatus helps in the formation of lysosomes. So there, Golgi plays, our dear Golgi It plays a great role for the formation of lysosomes. Now the structure. Every cell organelle, they will explain it. We have to start with the structure. And then you go on when we write. Remember the structure, when you write an answer, explain the structure. Or like for ER, you have the different two types, and then you write the structures, whatever role it plays. So every time when you write an answer for this, you draw, then you write the structure, and then you continue the important role or whatever functions it has. That is the way to write this answer. Lysosomes are again membrane. Everywhere you can see it's membrane bound sacs filled with digestive enzymes. Enzymes that help in digestion. And these enzymes, see a great role here. We have learned earlier that about the digestive enzyme. Here these enzymes are made by whom? Rough endoplasmic reticulum. So you got the all are related. That's what I said. They are all interrelated, but they have different functions. There are different types of cell organs, but they have different functions in them. That's what in the beginning I said. So lysosomes are membrane bound sacs filled with the digestive enzymes, and these enzymes are made by RDR or rough endoplasmic reticulum. You can see the membrane marked here. I hope it's clear for you guys too. And the other markings, you don't worry. I wanted to show you the membrane, and there's a lipid layer for the lysosomes. So this will be given in a simple format. You don't have to worry about it. Now here, I don't know, last time I've mentioned about lysosome, they are suicidal bags. In my class, I think the other classes also teacher must have mentioned, they are suicidal bags and all those. I think by now somebody saying, yes, we remember. It is this. Lysosomes are the waste disposal system of this cell. So very important. This cell organelle the lysosomes are the waste disposal system of the cell. Now what is the function? What do they do? It's understood when that waste disposal system, that word itself tells it out. They help to keep the cell clean by digesting any foreign material, any material getting from outside to inside, and worn out cell organelles, any organelles which does not work out. Okay, the old ones or which does not work and does not function well. So lysosomes play an important role here. Lysosomes are the waste disposable system of the cell, and they help to keep the cell clean by digesting any foreign material and worn out cell organelles. It's a function. Please underline, or we can note down this. This is there in the text. Why I'm stressing these lines are important. Are you with me, or are you sleeping? I hope you're with me. Okay. Now we're continuing with lysosomes. Foreign materials like what? Yes, somebody acknowledged that you are with me. Thank you. But now come back. Foreign materials like bacteria or food or old cell organelles end up in lysosomes. So foreign materials like bacteria or food or old cell organelles end up in lysosomes. At the end, they come to the cell organelle. What they do, which will break complex substances into simple substances. Then, lysosomes are able to do this because, why are they able to do this? Why only this? Because they contain powerful digestive enzymes capable of breaking all organic material. They can't just simply, one cell organ cannot just simply do this function. There must be a reason for it, there will be a factor for it, and that is they contain powerful digestive enzymes capable of breaking down all organic material. Got me? So this is important. Why is lysosome able to do this? How it helps them? Because they contain powerful digestive enzymes. Now, in case if anything happens or disturbance is caused in the metabolism of the cell, what will happen? In case there's any disturbance in metabolism or the cell gets damaged, again I'm saying, if anything, any disturbance is caused in the metabolism, in the working or the biochemical activities happening in the cell, or if the cell gets damaged, lysosomes may burst off. They just burst, and the powerful enzyme digests its own cell. Okay? This is important. In case if there is any disturbance in the metabolism or the cell gets damaged, lysosomes, they don't tell us, they burst, and the powerful enzymes digest its own cell. That is why, or therefore, lysosomes are known as suicide bags of a cell. This is important. Why? If there is any disturbance in the metabolism or if the cell gets damaged, they don't wait. Oh, please wait, I'm just going to do this. No. They just burst. This lysosome just burst. And the powerful enzymes digest its own cell. Therefore, lysosomes, there's a question as such for this I'll be giving. Lysosomes are also known as suicide bags of a cell or suicidal bags of a cell. So which organelle is known as suicide bags of a cell? You must be ready to answer it. That is lysosomes. Okay, don't worry. This won't be taken now. So this I just want to know. This is my favorite. I know my guys who were there last in my class. Teacher's favorite. She was always behind this mitochondria. Powerhouse of this. She was always with this. In fact, all the teachers, not only me. Okay, next class we will be going through mitochondria. So we have done with the nucleus. Please go through that. It's vast about it. When you saw on the slide, it looks easier, but it is pretty big. We have done with the nucleus. A short we've given what about cytoplasm. Now we are done with the new cell organ, the endoplasmic reticulum. Okay, very important. Okay, then we are done with our dear Golgi Paratus. Okay, and then after that we are done with our lysosomes. Mitochondria, my dear favorite mitochondria, the powerhouse of this cell. My favorite. You know, some of this saying, yeah, teacher's favorite. Always she used to say last year, mitochondria. Yes, it is my favorite, okay? Now, this will be taken in the next class. Now, I just want you to take, if you have your text nearby, just take page 60. This chapter, Fundamental Unit of Life, take page 60. I want you to see, to understand about hypertonic, isotonic and hypertonic solution. The easier way is there to understand. So you just go through this activity and I want you to, page 60. I hope all of you have taken that. Activity 5.3 and 5.4. So, some are still struggling, I can see. Where? Did you take it? Page 60. Some are just looking at me, smiling, swastika, smiling to glory, no text with you, darling. Swati is also, Swati, Sri Kumar, smiling. Oh my God, she's blushing. Prashant, open the text. Are you ready? There, he's also smiling. Oh God, teacher's calling my names. Okay, Navneet. Good. Oh my God, he's on spot, he's on time. Okay, my dear children, page 60, activity 5.3. Go through that. That is an activity, but hydrochloric acid is mentioned. Don't worry, my God, there's no hydrochloric acid at home. You don't go and run for that. Go through that. You'll understand exactly what we have mentioned. 5.4 is an activity you can do. You can do that. You have that at home. Otherwise, you somehow arrange that. Raisins, the dried grapes. Okay, I'll tell you what is it. The ones which you use for kheer or the paisa, those. Get 1 or 2 or maybe 3-4 pieces. I'm sure it will be in all of your houses because when COVID started, all mothers are busy. They have become great chefs. So they all are ready with the spice and cake and so I'm sure that is there in the fridge. You better. I do have. I was with this, enjoying. Though now I miss you guys. So you take that and do that activity 5.4. You will understand clearly. That is the only example you can understand very clearly what are those solutions and what is happening in it. I hope you will do, and I'll pick in the next class to ask, did you do or not? Or I'll tell you to take the picture and send it to you on my WhatsApp. I'll pick up random selection, okay? Okay, now I want you to switch on your videos and I wish to see all your beautiful faces.